So my name is Veronica, and this station is all about DNA, deoxyribonucleic acid. So we first want to introduce how DNA is made. So that's why we have some sweets here, and we use them to introduce the idea of base pairing and the phosphate backbone. We then introduce kids to the idea of single nucleotide polymorphisms and how DNA can be used to identify criminals. So they use a conversion chart to identify the suspects or the criminal that's committed the crime by stealing some exhibits from the Hunterian Museum, and that tells them how single nucleotide changes can introduce differences in the physical way of looking, and then that gives them an idea about the next stage of the activity.
So this is a molecule of DNA, and it tells us what the body's supposed to make and what the body's supposed to do. So firstly, the first stage is we're going to make some DNA from some sweets. So as you can see here, these two red strands on either side, these are the sugar phosphate backbones, and then in between we've got some some gummy bears, and these pair together to make base pairs. So basically that's the first stage, is we're looking at what the DNA is made of and how it's made. So then we move on to the second stage, which is your bit.
So this is where we identify the criminal based on so we instead of saying like nucleotides, we can say a single change in the gummy bear can mean that there's a difference in our hair color or eye color. So then they compare the DNA of the suspect or the criminal identified with our conversion chart, and then they look at the photographs and say which one of these they think is the criminal, and then they pick up the box and it tells them whether they were correct. And then they get a sticker and then they move on to the next stage.
So this is like the real world application of like understanding the DNA with these like single letter changes. So this is a problem. The Scottish wildcat is the most endangered mammal in the world, and there's only thought to be about 35 left, and they're going extinct due to crossbreeding with non-pure wildcats. So there's a couple of different conservation strategies, and one of them is based on catching the cats, testing their DNA, and then being able to tell which group they fall into. So the Wildcat Haven they have sequenced both feral cats and wildcats, and they've found 63,000 points in the genome where there's different letter changes. And so using this, they're able to catch cats, test their DNA, which will tell them whether they're a wildcat, a domestic cat, or if they have a kind of mix of both, then they're going to be a hybrid. So this activity, so you start off, it's just a wee bit of background information to let you know about what's happening. You take a wee investigator logbook, this is for all your answers. And then the first thing that you do is you separate the cats. So you've found 8 cats, and you've got to separate them just based on what they look like. So we give you here the characteristics that are associated with pure wildcats and domestic cats. And using this, you've got to decide whether these cats here that you found are domestic cats, hybrid cats, or pure wildcats. So that's your first set of answers, which will go in here. But then you manage to actually test their DNA. So then you're establishing whether, looking at their DNA, do you get different results as to when you're just looking at their phenotype. And it's kind of hit home that even though a cat may look like a pure wildcat, when you actually look at this DNA, it's actually a hybrid cat. So for the conservation project, you need to be able to tell which ones are pure wildcats so that you can neuter them, so only kittens you should be getting in this safe haven are going to be pure wildcats. And then if they get everything right, we have this wee photo thing. Do you guess what I'm going to do? And they get to become a wildcat.